everyone, and welcome back to Frontlines of Future Care. I'm Samira, and today I have the pleasure of being joined by Professor Martin Taylor. Marty Taylor is an assistant professor at Brown University whose work lies at the intersection of biology and technology. I'm so excited to dive into this conversation. So, Marty, thank you so much for being here.
Pleasure.
So, to start us off, I would love for listeners to get a big picture idea of what you're doing. How would you describe the work you do at the intersection of bio and tech?
Sure. So I'm a physician scientist and gastrointestinal pathologist, and we work on something that used to be called junk DNA, which is a line one transposon, which makes up a huge portion of genomes and is increasingly recognized to be a contributor to human disease. And we're trying to figure out how much it matters and can we target it for diagnostics and therapies. And so we kind of are unusual in that we do everything in the lab from single molecule cryo-EM protein structure determination through translational work into people with diagnostics and trying to run clinical trials and drug development. And so we use a variety of different tools and collaborate with a lot of teams in sort of a team science approach. And so I think where we use AI the most, which I think is what this audience is interested in, is around protein structure prediction and around literature mining in areas that are new to us.
Right,
Um, so I think I can be a helpful, especially there.
right, yeah.
And then we also use a lot of we use machine learning in evolutionary analysis as well, mostly in collaboration with the Greenbaum Lab.
Nice. So yeah, so you talk about using these protein predicting tools. Can you walk us through how you use it in the space?
Yeah, and it's been a huge revolution. I think anybody who's looked at this space is aware that, AlphaFold, when it came out a few years ago, was orders of magnitude better at protein structure prediction than any previous algorithm. This is a space that I've been working in since around 2005 as an undergrad. And the both from the structure prediction and the protein design side, everything has really been revolutionized by machine learning over the last couple of years. Um, I kind of got into the space because of some of the initial work from the Baker Lab, for which David Baker actually recently won a Nobel Prize. The idea that you could predict a new protein fold or
Mhm.
design a new protein fold and then actually make it happen. Um, but so, what's happened is that the accuracy has gone up really dramatically.
Mhm.
And for example, when AlphaFold first came out, we and many others had crystal structure data that we hadn't yet finished fitting and solving. And overnight, the predictions from AlphaFold were essentially just right and close enough to right that we could actually solve our experimental data. And that was really transformative for us and for many people in sort of the drug discovery protein structure space.
Mhm.
And then, very quickly the question started being, why are we even doing this if the computer can predict it? And what can we do that the computer can't do? And
Right.
anybody who actually did this work knew that that was crazy because the computer is only as good as what we had previously seen, and things are much more complicated than this.
Yeah.
So, we have found the tools to be extremely useful, and we're also like very acutely aware of their limitations.
Right.
So what we are interested in how these very large macromolecular assemblies work and how proteins, especially in the transposon, it's essentially a virus, and how this virus enzyme that's evolved to be this ultra streamlined parasite over like 2 billion years, essentially,
Mhm.
how does this very specific machine work? And so what the computer was able to do, what the model was able to do almost immediately is tell us one state of a three-dimensional structure of it. And it was able to predict some of where the DNA and RNA would bind to the protein. What it can't do correctly, it can't get that second part right. So the DNA and RNA bind in a couple of different places and then moves through a cycle where it starts over here and then it pushes it through and it
Mhm.
ratchets it along and then it wraps around. And all of those predictions um, were not really great.
I see.
Um, and then, the other piece that we're very interested in is to know what it interacts with and where. And we've done a lot of work on this. And One thing that was very interesting is that AlphaFold basically showed that it predicted that the model that we had was wrong. We had predicted based on sequence that the protein PCNA would interact, which is it's a DNA clamp. It's a
Mhm.
three-piece ring that is loaded onto polymerases. We had predicted that it would bind to a thing called a pit box, which is the sequence motif that other people had identified. And we mutated the pit box and we were able to break binding. But what our mutants actually did, it turns out, was break the protein and not break the interface. And AlphaFold predicted that it didn't sit on the back of the protein there, it sat over here instead on the front.
Mm.
And it turns out that that looks like it was right.
Oh,
Um, and so, sometimes it can be really helpful in that way. But the other thing that it does right now is if you just say, look, show me how these two things will interact, it makes up an interaction.
right.
And it'll tell you if it thinks it's right or not with some confidence, but at the end, it just doesn't really know. So
Right.
when you start to look at large protein complexes and how they come together, the predictions are helpful if you have high quality knowledge ahead of time that there's
Okay.
likely an interaction,
Right.
but it's not good if you're fishing right
I
now.
see. Yeah. So that kind of leaded to my next question. Obviously, there's a lot of ways that these tools can help us, but I was wondering if you ever see these technologies being misused, or if you think that, it's just maybe more beneficial to not even use the tools in the first place, but kind of just work together with, actual humans instead of AI.
It's always better to work with actual humans who know things than
Okay.
AI.
Okay.
No, that's not what your AI people want to hear, but
No,
a
no,
lot
we
of the
have different
AI is
perspectives.
still garbage in, garbage out. And it's getting better, and sometimes it's very useful. But the, the like for literature mining and knowing what's important, sometimes AI can be great, right? We're moving into a new area. We're working on a cytokine we didn't used to work on. Want to know what it's been done with this particular cytokine in cancer. And it'll pull three studies that are close to right, although the links are often not right. So I'm talking now about, for example, like a ChatGPT type model, their,
Hmm.
pay version that's the fanciest, newest, research only one that is quite expensive to use. But then it just lies. Like it told me, for example, that a clinical trial done by a colleague of mine tested whether this cytokine was effective in addition to immunotherapy in cancer. And of course, the trial had nothing to do with the cytokine. It was just an immunotherapy trial. And it just
Okay.
made it up. It
You're
just
right.
made it up. And so that kind of hallucination, I know people are talking about it a lot. It's still a big problem. And so we're very much on the trust but verify for any of this
Right.
kind of stuff.
Yeah. That makes sense.
But it can be really helpful to find a useful paper that you hadn't found otherwise and to go to the papers that seem important rather than the ones that match your keywords the best.
Yeah, definitely. Yeah. So looking ahead in this changing field, where do you see the biggest opportunities for digital tools to transform protein biochem in the next 5 to 10 years?
Yeah, I mean, we spend a lot of time trying to figure out how to make proteins and figure out what they do. And so, new tools to predict what is going to be able to be made and what isn't, what's going to be
Hmm.
aggregated, what systems we need to use, what might work, how to do it. We spend a lot of time with sort of it's kind of alchemy, I don't know, where, some combination of experience and trial and error
Hmm.
to try to actually figure out how to get cells to make enough of a protein that we can study what it does. So I think as if that data is integrated, because most of what happens is people fail a whole bunch of times and then
Hmm.
it works. And then the thing that works gets published in very, very short brevity without a litany of all the things that didn't work in biology. And that's been the culture for a really long time is that nobody really cares how hard it is. People just want to know what the biological phenomenon is, and the technical part is often what people spend their time on, but not what they talk about. So, so if people could find ways to get that data or to think about that data in a more holistic and global way, it would really speed up a lot of research efforts because you would skip the failures.
Yeah.
Um, or at least reduce them. There's going to be no way that everything works the first time, even when you do the experiment right.
Yeah.
But
No, that perspective, that's really valuable. Yeah, so on the flip side, as this data gets more complex and we're moving forward, is there anything that researchers need to do differently in the next 2 years?
I mean, I think people are learning really quickly that there are places where we can save time
Hmm.
using these kind of models. Um, but ultimately, a lot of what the business that we're engaged in on the academic side is also training new scientists
Hmm.
how to think and how to design experiments and how to ask incisive questions. And right now, I really don't have a great sense of how we can, because people are trying to start to use these models to learn and to guide their reading and things like that. But um, yeah, I think we're going to need to figure out as a field how we train people and how
Hmm.
we teach students, how to use these kind of tools. And I mean, what's been really fun is to see people coming up with new approaches with these
Right.
kinds of tools for structure prediction, for predicting how a protein will bind to some nucleic acid sequence, or what's the motif that this thing recognized, where is it going in the genome,
Hmm.
all these complicated pieces of data. Um, and sometimes the answer is just like it just didn't work, and people we have to figure out how to be critical of that and how to be open to being critical that things just,
Don't work.
aren't work
Yeah.
aren't working. And so we're trying to be incisive in that way and trying to teach people to think like that. And then
Awesome.
when it works, then to embrace it and figure out what you learned.
Yeah.
But at the end of the day, most major biological discoveries are something simple. And so when a lot of really complicated data turns into a story, it's great. But a real biology paper is we did some fancy approach and we learned a thing, and then we showed you in 3 or 4 simple ways that the thing is true and
Hmm.
why it matters.
Right.
Right. And so where we can use these approaches to find something we didn't know or to give us some insight as to, you know, what is or isn't working or why some patient population is responding in some way and another one isn't, I think that'll be helpful.
Yeah, definitely. Okay, so a fun question at the end here. What's the most surprising fact about biochem, cell biology that you wish more people may know?
So your genome is only about 2% genes, but
Okay.
about half transposons, mobile DNA,
Mm.
which are these virus-like sequences that just encode their own copying mechanism and just copy and paste themselves over and over again. So you're actually like far more virus than human.
Ooh,
Your genome, yeah, your genome is probably 25-fold more virus DNA than human DNA.
wow. Got it. That's very interesting. Okay, so thank you so much for joining us today, Marty. I know our listeners will find this conversation very interesting, just as I did. So thank you once again.
Pleasure. Great to be here.